Hi, in this video, I want to show you how I teach my students some difficult to grasp concepts, how I answer some of their questions, and address some misconceptions. So the video is about cellular transport, the semi-permeability of the membrane, and specifically diffusion and osmosis. And what I'm showing you here in this model is a model of the cell membrane. You can see the phospholipid bilayer, cholesterol molecules within the bilayer, glycoproteins, glycolipids, channel proteins, and all kinds of other proteins, surface proteins, integral proteins, peripheral proteins, and so on. I took this image from a website called biorender.com, and you can, using that website, you can actually make any model you want, any drawing you want, and it's beautiful. It has lots of pre-made shapes that are labeled. And in this case, I was really lazy because I took a template that was already made by someone and I just modified it. So what I added actually was this molecule of oxygen here in this arrow, and this molecule of CO2 here in this arrow, and water molecule. So you can find all these shapes on the website. You actually don't need to make them. So why am I showing you this picture? One of the concepts students struggle with is, well, how do the molecules pass through the membrane, right? They know that small nonpolar molecules pass through the membrane, and they know that they pass freely through the membrane because they're small and because they're nonpolar, right? And the tails are nonpolar. So tails are nonpolar and hydrophobic, and the molecules are small and nonpolar and hydrophobic. So likes dissolves like, so they can pass through the membrane. But then they get stuck up on this. Well, the heads are actually polar because they're facing water and they're charged. So how come nonpolar molecules can pass through the heads? Because the membrane is not entirely nonpolar, right? And then some students also ask me, but well, even if nonpolar like each other and get dissolved, how do the molecules actually traverse the membrane? How is that possible? So they know it, but they're having a hard time visualizing it and understanding it. And so this is what I tell them. Now, imagine each tail of the phospholipid is actually a long hydrocarbon chain, right? It could be anywhere between, I forget now, 9 and 20 carbons. 9 and 20 carbons. So between 9 and 20 atoms. And we're not even counting the hydrogens. Now, an oxygen molecule has only two oxygens, so two atoms, versus between 9 and 20 atoms. 3 atoms for carbon hydrogen, for carbon dioxide, versus between 9 and 20. So the molecules here that I show you are not really drawn to scale, because if I make them as small as they actually are compared to the tails, we won't be able to see them. So I make sure I tell my students that they're really, really tiny compared to the tails. That's one. Two, we talk about the membrane as being mosaic and fluid. And I remind them that fluid means liquid. Liquid means molecules are in constant motion. So the tails are actually moving. They're constantly moving. The proteins within the membrane are also moving, but the tails, the phospholipids, are in constant motion. And as they're moving, they do create small openings through which small molecules can sneak through, literally sneak through. And my students look at me as if I'm crazy, but then I show them this. So this is a bead curtain that some people have over their doors. And if there is wind or if there's like people coming in and out, they're constantly, these beads are constantly moving, right? They're in constant motion. And you can see right here in the picture that some of the beads are closer to each other than others. And there's these tiny little gaps in the bead curtain through which small molecules can sneak through. And when I show them this, they can really get how small molecules can sneak through the membrane. Now, these openings allow small molecules to sneak through, right? But if you are polar like water, you're going to have a hard time being in this space right here where the tails are, right? Because the tails are hydrophobic. So you're kind of going to be stuck there. But if you are small and hydrophobic, like oxygen or carbon dioxide, you're non-polar, you're going to have a much easier time traversing this distance. This distance is large, right? The two tails. 20 on this side and 20 on that side, that's 40, right? So that distance is much easily traveled through if you are non-polar, if you're hydrophobic, because the tails are hydrophobic. But if you're small, you can see through the small openings. Sorry, apologies. You can sneak in through the small openings of the membrane. Whether you're water, whether you're oxygen, whether you're carbon dioxide, you will sneak in. Problem is that if you're polar, you will kind of be stuck in here and you will not move very fast. But if you're non-polar, you can move quite fast. So oxygen can freely pass from the extracellular space into the cell. Here's the arrow, right? Because we get oxygen because there's more oxygen outside of the cell in the blood than it is inside of the cell. And carbon dioxide, which accumulates in the cell as a product of cellular respiration, it can now leave the cell because there's higher concentration inside the cell than outside of the cell and passes freely through the membrane. So this is how I explain to my students how small molecules pass through the membrane. So they can all pass because they're small compared to the large phosphate heads and the large tails. And because the tails are in constant movement, the phospholipids are constantly moving, they can sneak in through these tiny temporary openings. I tell them those openings are temporary, just like the curtain. It's this space between the beads, or this space that the girl now is making, is only temporary. They're going to get back together. So these are small temporary openings through which all small molecules can sneak in, right? But once they've snuck in, if they're non-polar and hydrophobic, they can easily move through the length of the phospholipid tails because the phospholipid tails are non-polar. But if they're polar like water, they're going to kind of be going very, very, very slowly because they're going to be stuck in this hydrophobic phospholipid bilayer. And for that reason, the majority of water moving in and out of our cells moves through aquaporins, right? These special protein channels that are designed to move water. So that's one thing I use to explain to my students how molecules pass through the membrane and why the membrane is really semi-permeable. Now, of course, as the molecules are larger, like proteins, or larger lipids or large carbohydrates, they will not be able to pass at all because they will not fit in through these small temporary openings. So there is no passing if you're a larger molecule. So you will need the help of a protein channel or a transport protein to enter or exit the membrane, which means automatically that the cell decides what goes in and what comes out of the cell. So this is the simplest way possible I can think of explaining to my students how cellular transport works. Okay. So the next thing that students often struggle with is net diffusion and equilibrium, something that we call actually dynamic equilibrium. So net, I always tell them, net means when you add the molecules that move to the right and subtract the molecules that move to the left, and you get a positive number, that means molecules are moving to the right. So the net movement is to the right because there's more molecules moving to the right than they are moving to the left. Sometimes I tell them when you go to the store and your mom and dad buy hamburger or something, they put the person in the store puts a piece of paper where they're going to wrap the meat cuts on top of the scale. And then they're going to tear the scale because they want to subtract the weight of the paper from the actual weight of the meat, because you don't want to be paying for the paper, you want to be paying for the meat. So that's called net weight. So that's the total weight of your package minus the packaging is the net weight. That's what you're paying for. Same thing with diffusion. Molecules are moving in both directions, Brownian motion. They're in constant motion, right? If most of the molecules are moving to the right and few are moving to the left, then the net direction of movement is to the right. Once they understand this, And only once they understand this, I go to equilibrium and I tell them that equilibrium is really dynamic. It's not static. Molecules are still moving both left and right. But in an equilibrium, if 10 molecules move in, 10 molecules move out. And the same number of molecules remain on both sides. And I use another analogy by saying I have two sections of AP. Each section has 18 students. They're one right after the other, right? So the bell rings, 18 students leave my classroom, 18 students come in. The next class comes in. So there is no net movement of students because, yes, 18 left, but 18 came in. So there's an equilibrium in my classroom. Okay. So again, it's important first to stress that molecules are in constant movement and that net diffusion means this is the direction in which the majority of the molecules are moving, but it doesn't mean there aren't some that are moving in the opposite direction. And only then can they understand equilibrium. So equilibrium means that equal number of molecules are moving left and right. So overall, the net movement in equilibrium is zero. That doesn't mean the molecules are not moving, it's just that net movement is zero at equilibrium because as many molecules move to the left as they are moving to the right. So that is another concept that students sometimes struggle with. Okay, now we're going to switch gears and talk about osmosis. And I'm sure you've all seen this figure of the YouTube, the original YouTube, where you have a YouTube filled with water and sucrose, in this case sugar, because in the same permeable membrane separates the YouTube in two, and the sugar cannot move through the membrane. So when the solute cannot move, then what is moving? The water is moving, correct? And we can determine water movement by the fact that in the beginning of the experiment, the height of both tubes is the same. At the end of the experiment, the height of one of the tube is higher because water, and the other one is lower because water has moved from the left tube to the right tube. And why is water moving from the left to the right? Because there's more solute, there's more sugar on the right. There's a high concentration of solute on the right than on the left. So water is going to be moving to the right, and you will see how the volume of the right increases, the volume to the left decreases. So I don't know whether you guys teach hydration shells. It's technically not in scope, but I think it's important for students to understand this, and I'm sure they've had it in chemistry, that the way water dissolves polar molecules is by forming hydration shells. And you can see it here in this figure, how here's the molecule and it is covered in water molecules. So the solute molecule is covered in water molecules. So the water molecules form a hydration shell. And I also tell my students is like the water is hugging the molecules it's dissolving. And when my students have no issue with this, I typically go around the student, put my hands in a circle around the student to show them how water forms a hydration shell around a solute molecule. Okay, the net movement of water across the semi-permeable membrane we call osmosis. Okay, so and I want to skip this for a second. Osmosis is important, especially important in cells that have a cell wall. We typically say plant cells, but those are not the only cells that have a cell wall. Bacteria and fungal cells also have a cell wall. And students have to learn about water potential. Okay, so the first thing I do is I introduce the concept of pressure potential. And this is one of the best ways to introduce the concept of pressure potential. You show them the same YouTube, right? They already know what's going to happen, what is going to be moving up the right hand tube because there's more solute on that side. And the volume of the water, of the, the volume of liquid in the right hand U-tube is going to increase. So which way is the water moving? If you think about the tube, just the vertical part of the tube, the water is moving up, correct? Because it's moving from the left to the right, all the way up, right? So you see how the volume here, the height of the liquid in the right hand tube is higher than the height of the liquid in the left hand. So the water has moved up, right? The blue arrow shows you water movement. What is pressure potential? Pressure potential is a force that goes in the opposite direction. If your students have had physics, you can invoke Newton's laws, right? For every action, there's a reaction. So it's a force going the opposite direction. So I show them a plunger, like from a syringe. You can even use a syringe to demonstrate it, because I doubt we have YouTubes in our classrooms. And imagine the plunger going downward. See, the red arrow is showing you which way the plunger is going. So the plunger is going in the direction opposite of water movement. And it will prevent, if you were to use a plunger, without a plunger, maybe the liquid is gonna come all the way here. With a plunger, the liquid may not go all the way to the top. It's gonna go only up to here, because the plunger is exerting pressure and preventing the water from moving all the way it could. That's what I tell my students. So the water, if you let it, it can move until the end of days. Well, not really, but I exaggerate. But the plunger exerts pressure and will prevent the water to moving as much as it would like to. So this is how I introduce the concept of pressure potential. Okay. And then I show them this picture, which comes from, I actually showed them the whole video from Mr. Anderson's video, which is excellent. So if you have more solutes inside the cell, your solute potential is very, very negative, then water is going to be moving in, the green arrows, into the cell. Now, if it were an animal cell, the water is going to move in until the concentration on both sides of the membrane are equal, or the cell bursts, most likely before the cell will burst first, rupture the membrane, if a cell is placed in a hypertonic solution. However, in a plant cells, the water movement will stop, and it will stop because of pressure potential. So when enough water has entered the cell to exert pressure, the pressure potential will counteract, just like this plunger, the movement of water. So remember, water goes up, plunger goes down. Same thing here. The green arrows mean water goes in. The white arrows mean the pressure potential exerts force on the cell wall in the opposite direction. So it will prevent, it will slow down the movement of water, and eventually it will prevent the movement of water. So I show them this so they can visually picture what is going on. They need to imagine it. They need to see it first before I throw the formula at them, which is psi equals psi s plus psi p. Okay. But this really helps to show them something visual. And this is one of my favorite figures, which explains it in a different way. A beaker with distilled water has a water potential of 0 because it has no solutes dissolved. So the solute potential is 0. There is no water pressure potential because it's open to the atmosphere. So 0 psi p plus psi s equals 0. Now we are going to place a plant cell inside a beaker with distilled water. Now, obviously, the plant cell has a water potential that's different than 0 because what? It has a solute potential. There are all kinds of solutes inside the cell. And let's say the solute potential is -2. And we're going to get back to why solute potential is always negative in a few minutes. So solute potential is -2. Right now, there's no pressure potential. And so overall, the water potential is -2. So we place a cell with a water potential of -2 in a beaker with a water potential of 0. What's going to happen? What is going to enter the cell? Because water goes from high water potential to low water potential. So water potential measures the ability of water to move, right? And it always goes from high water potential to low water potential. So as water enters the cell, pressure is going to build up. And you can see how the cell looks like before water, and now it's like an inflated balloon because water has entered. As water enters, the pressure is going to increase, and now we're dealing with a pressure potential. And when the pressure potential becomes 2, remember the solid potential is -2, what's going to happen? Water movement is going to stop because +2 -2 equals 0, and now the water potential of the cell is 0, and the water potential of the cell equals the water potential of the beaker. Net water movement stops when the water potential of the cell is equal to the water potential of its surroundings. Okay. Now for animal cell, we don't talk about pressure potential because pressure potential is not does not exist. And we don't need to talk about solute potential. All we need to talk about is solute concentration because that's the only thing that determines water movement. But for a plant cell, for a fungal cell, for a yeast cell, bacterial cell, pressure potential matters. So that's why we use water potential to predict water movement. And if you think that water potential is not important, think about this. Only animal cells do not have a cell wall. The majority of the living world has a cell wall, some form of cell wall. So water potential is the only way really to predict water movement for every type of cell except for animal cells. And it is extremely important because this is how land plants actually get water all the way from their roots to the top of the plant. And we have actually simplified the equation for students by saying it's only important pressure potential and solute potential. But when plant biologists calculate how water moves up the tree, there are other variables, other potentials that actually influence the overall water potential. So all I'm trying to say is that sometimes it's important to explain to students, well, why we're talking about water potential and why are we looking at these formulas. There is a purpose to it, not just to make your life harder. Okay, so let's discuss one thing that I also use to tell my students. Whenever we give them formulas, I always tell them formulas are not to be remembered, okay? You're going to see that formula on the formula sheet. It's always there for you. No need to stress out. And even if you don't have a formula sheet later in college, formulas are not there to be memorized. They're there to be understood because formulas are models. It's a mathematical representation of a phenomenon. So it's a form of model. It's used to describe a phenomenon. It's used to predict effective disruption and so on. So this is the formula of the solute potential of a system. Psi S equals -I CRT. And we tell them I is the ionization constant, C is the molar concentration, R is the pressure constant, T is temperature in Kelvin. Okay. So each water molecule can hug one thing at a time. That's what I tell them. So remember the hydration shells. A water molecule water molecules form shells, hydration shells around the molecule they're dissolving. So one water molecule can hug one thing at a time. If I hug you, I cannot hug anyone else. And I demonstrate with students that. So a water molecule will hug either a molecule or an ion. Each time a water molecule hugs something, it can't hug anything else, which means that the potential to hug is reduced. So water potential is reduced. This is how I explain why water potential is reduced when you add solute to pure water. So pure water has a water potential of 0. When you add solute, the water potential becomes negative, so it's reduced. Why? Because now the molecules of water will hug all the solute in the solution, and their potential to hug other things is now reduced. So I demonstrate kinesthetically. what water potential means. If I hug you, I cannot hug any other student in the classroom, which means my potential to hug is reduced. So water potential is reduced. Now, if you have more things to hug, there's lower solute potential. And I even tell them this. Let's say there are 4 teachers in front of you in the classroom, and there's 4 students. You can each hug 1 student. Great. But if there are 10 students, I'm sorry, let me just take this back the other way around. So there's like, let's say 50 parents and 4 students. 4 of the adults can hug 4 students, right? And then there's more adults left to hug more students should more students come in. But each time a student comes in, there will be less and less and less adults, let's say teachers, able to hug you, to hug students. Because once I hug 1 student, I cannot hug anyone else. So my water potential, my potential to hug is reduced each time you add more students, more solutes to the solution. So adding more solutes to the solution lowers the potential for hugs, lowers water potential. That's how I explain it to them in a kind of visual kinesthetic way. Now, if you have 1 molecule, I can hug 1 molecule. Let's say the molecule splits into 2 ions. Now I have to hug not 1 thing, I have 2 things to hug, right? So if you dissolve 1 molecule, it can be hugged by however many molecules, water molecules form the hydration shell. So you form 1 hydration shell, right? But what if that molecule, when dissolved, splits into 2 ions? Now you're going to need 2 hydration shells. You're going to need a hug from 2 water molecules. So the water potential is reduced doubly. For that reason, we have that I thing. So I need to multiply things by 2 if I'm dissolving sodium chloride, because it's not really 1 molecule of sodium chloride. It's 2 ions. It's sodium and chloride. So you're going to need 2 hugs, not just 1. That's why you need to multiply everything by 2. This is how I explain the ionization constant. And the last thing that I use for my students to explain to them why water potential equals the negative value of the solute potential. So the solute potential is always negative. Why? We always put a negative sign in front of it. Why? Why is it negative ICRT? And this is what I showed them. On the left, you have a solute that can freely pass through the membrane. Which way is the solute going to move? It's going to move from the outside, in this case, to the inside of the cell. Why? Because there's more solute on the outside of the cell than inside the cell. So diffusion goes from high concentration to low concentration, correct? Now look on the right. Now I have a pink or magenta solute. This solute is also, there's also more on the outside than in the inside of the cell. And the dotted arrow shows you the direction in which this solute would move if it could pass through the membrane or if there was a channel protein. But it can't, okay? It's important to stress that solute cannot move. And because the solute cannot move, we need to concern ourselves with water movement. Okay? Because the solute cannot move, what is going to move? Water is going to move. In which way is water going to move? Water is going to move from the inside of the cell to the outside. So I want you to notice the direction of water movement. The direction of water movement is opposite that of solute movement. If the solute were able to move, like it is on the left, it would go from out to in. So it will go into the cell. The solute cannot move, so water goes out of the cell. So the movement of water and the movement of solute is in opposite directions. And how do we show this in our model called a formula? We show it by putting a negative sign in front of the solute potential calculation. Once I explain to my students, they never forget to put a negative sign in their calculation. So the negative sign sign in the formula of solute potential is there to reflect that water and solute will move in opposite directions. Okay, and I hope this was helpful.